Hello doctors. Welcome to Note Official. Let's break down medicine into concepts. Today we are going to talk about embryology week 3. In the last video we covered week 2. By the end of week 2 we have implantation is complete. Extraembryonic mesoderm is formed. Chorion and amnion formed. Primary and secondary chorionic villi formed. Amniotic sac, yolk sac and bilaminar disc present. And now we will move forward. We have epiblast cells and we know that they are columnar cells and hypoblast cells are cuboidal. Amniotic cavity is above the epiblast. Yolk sac cavity is below the hypoblast. We should know the orientations of the embryo. This is the tail end of the embryo and we call it caudal. Opposite to tail we have head part known as cephalic. Upper side is dorsal. Lower side is ventral. Now in order to form embryo, epiblast cells start to proliferate from the caudal to cephalic direction. We know this proliferation as a primitive streak. Let's draw it by ourselves so we can understand it more. Let us assume this is embryo and this is caudal part from where epiblasts start to proliferate towards the cephalic part, so remember this primitive streak grows in caudocephalic direction. What can we see in the cross-section of the embryo? Let's see. So we have epiblast cells and hypoblast cells as we already know. In the primitive streak, epiblast cells will proliferate to the ventral side. But we have hypoblast cells of the yolk sac cavity on the ventral side, right? So what will happen next is that epiblast cells will replace the roof of the yolk sac cavity. What is happening here is, epiblast cells are proliferating ventrally and replacing the hypoblast cells. Now we have a layer of epiblast cells ventrally, and it is known as endoderm. A endoderm is a germ layer. A germ layer is a primary layer of cells that forms during the early stage of embryogenesis. A endoderm is the first germ layer to develop an embryo. And now as we know, ventrally proliferated epiblast cells form endoderm. But during the proliferation, some cells get stuck between the endoderm and the upper layer of epiblast cells, and they will form mesoderm. Mesoderm is the second germ layer. We can see here mesoderm, it is between the upper layer and the endoderm. So now we have endoderm and mesoderm, but some epiblastic cells are still there and they did not migrate ventrally. These cells will form ectoderm. Ectoderm is the third germ layer. Now we know that epiblastic cells proliferate ventrally to form 3 germ layers, and this process is called gastrulation. What is important here is the order of germ layer formation during gastrulation, and we all must know this. So the first germ layer is endoderm, second is mesoderm, and third is ectoderm. And now let us see in 3D how it works. So here we can see from the caudal to cephalic part in the primitive streak. When epiblast cells proliferate ventrally, replace hypoblast cells and form endoderm. Cells between endoderm and epiblast form mesoderm. And cells that did not migrate will form ectoderm. We have to think of this whole process in 3D. We can't draw in 3D, but we can assume it like this. We have the innermost layer endoderm, middle layer mesoderm, and outside layer ectoderm. If we take a cross-section from the embryo, ectoderm is outside, endoderm is inside, and between them we have mesoderm. And now let us see in the diagram and move forward. Let's assume we have cut a slice from the embryo as it is growing. We can see here 3 germ layers in order from outside to inside. And we can see inside we have endoderm and it is like a cavity. So now think, what do we have inside the body? The gastrointestinal system, right? So endoderm will form the gut tube in the future, and we have 3 parts of the gut: foregut, midgut, and hindgut. The middle layer is mesoderm, and it will form the cardiovascular tube like the heart, smooth muscle, and skeletal muscles. Ectoderm will form the neural tube, and it contains the brain and spinal cord mostly. So as we know, this is only for our understanding, but in reality it is like a ball. Assume we have endoderm inside, then mesoderm, and outside ectoderm. So we have the gut tube inside around that smooth muscles and vessels, and then the brain, but we don't have the brain outside, right? We will understand how that will happen. So let's move forward. We have the primitive streak here, but now it is ectoderm. Now let's draw it and see what will happen next. This is ectoderm, endoderm, and in between is mesoderm. And in the ectoderm we can see something like a groove from where cells migrate, right? And on the surface we have another part of ectoderm. We will call this groove neuroectoderm, and the ectoderm on the surface is surface ectoderm. Here we can see it in the diagram. This is neuroectoderm and this is surface ectoderm. Here we can see the junction between the two ectoderms. There will be formation of neural crest cells. Neural crest cells is the fourth germ layer between the two ectoderms. And now, as we saw earlier in this diagram, and we can see endoderm is forming foregut, midgut and hindgut. Here we can also see ducts coming from the midgut and hindgut. The duct from the midgut is known as the Vitelline duct. And from the hindgut is the Allantois duct. Here we can see the heart and vessels, and muscle from the mesoderm. And this ectoderm will form the brain here and the spinal cord. And here have an eye. This is the umbilical cord. Now, let me remind you, before gastrulation we had epiblast and hypoblast here. So what happens before gastrulation is, from the caudal side epiblast cells, primordial germ cells migrate before gastrulation at week 3. And they migrate to the endodermal part of the yolk sac. And we know the yolk sac is connected to the gut tube. So they will travel through the Allantois duct and go to the hindgut. From the hindgut they will reach the gonads at week 5. So this process starts at week 3 and takes 2 weeks for primordial germ cells to reach the gonads. At week 5 primordial germ cells are reaching gonads. Primordial germ cells are the first derivatives of epiblast cells. Let's make it clear before we move forward. From the epiblast, primordial germ cells are the first to form, meaning they are the first derivatives. Then endoderm forms, which is the first germ layer. Then mesoderm forms, the second germ layer. Then ectoderm forms, the third germ layer. And at the junction of ectoderm, neural crest cells form the fourth germ layer. Let me tell you one thing about primordial germ cells. If they do not migrate to the gonads but instead go to the tail of the embryo, it will cause sacrococcygeal teratoma. And if they go to the oropharynx, it will cause oropharyngeal teratoma. I hope you all understand week 3 properly. The next video will be on week 4, so subscribe the channel so you won't miss out. Before finishing, let's solve the questions. When do the first derivatives of epiblast cells form? Option A, week 1. Option B, week 2. Option C, week 3. Option D, week 4. Comment down your answer. What is the direction of primitive streak growth? Option A, cephalocaudal. Option B, ventrodorsal. Option C, dorsiventral. Option D, caudocephalic. If you want to solve more quizzes, follow us on Instagram, NoteNoteOfficial. Thank you for watching. See you in the next video.